Chapter 10 covers lipids. In this chapter, we're going to discuss the biological roles of lipids and the structure and properties of storage lipids, membrane lipids, and signaling lipids. Lipids represent a structurally diverse class. You have some that contain fatty acids, which have a long hydrocarbon chain and a carboxyl group. You have some that have a steroid nucleus, which consists of rings fused together. But importantly, they are all characterized by low solubility in water, meaning they are relatively hydrophobic. And that's due to all of the carbons and hydrogens in the molecule and very few oxygens and other polar groups. Lipids serve a lot of functions. They are useful for the storage of energy, insulation from the environment, water repellent, and they actually can help with buoyancy control and acoustics in marine mammals. They are important in cell membranes, and they can act as cofactors for enzymes. They are signaling molecules, pigments, and antioxidants. Here are two examples of lipids that act as pigments. Compounds with long conjugated systems. So conjugation means you have alternating double bonds, and there's resonance. Absorb light in the visible region of the spectrum, and subtle differences in the chemistry of these compounds produce pigments of strikingly different colors. Birds acquire the pigments that color their feathers red or yellow by eating plant materials that contain these pigments, and the difference in pigmentation between male and female birds are the result of differences in intestinal uptake and processing of these compounds. There are two major categories of lipids: lipids that contain fatty acids and lipids that do not contain fatty acids. And then the first category can be further separated into two types: storage lipids and membrane lipids. So let's first talk about fatty acids. A fatty acid is a carboxylic acid with a long hydrocarbon chain that contains anywhere between 4 to 36 carbons. Almost all natural fatty acids have an even number of carbons, and most natural fatty acids are unbranched. There are three different types. You could have saturated, monounsaturated, or polyunsaturated. With saturated fatty acids, you have no carbon-carbon double bonds. With monounsaturated, you have one double bond, and with polyunsaturated, you have more than one double bond. There is a particular fatty acid nomenclature that's used. There is a systematic name where you indicate that it's a fatty acid by the OIC ending, and the number of carbons you indicate here. And then you indicate the placement of the double bond with this number, and then you also indicate if there is a double bond, whether it's cis or trans. So there's the systematic name, there's also a common name, and then there's the delta and omega numbering system. In the delta numbering system, you start from the carbon of the carboxyl group. So let's look at this example. The carbon of the carboxyl group would be 1, and then you number left to right, 1 to 18. The location of the alkene you're going to indicate by using the carbon number that it starts at. And then you want to illustrate that it has a total of 18 carbons with this notation. So you have the total number of carbons, 18, colon, then the total number of double bonds, which in this example would be 1. And then the delta 9 tells you that the double bond starts at carbon 9. Then you have the omega numbering, which goes in the other direction, where you start from this end of the fatty acid. And in this direction, you would number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So in this particular example, you also get 18 carbons, and then the double bond would also start at 9, numbering from the other side. But instead of using the delta symbol, we're going to use the omega symbol with the number 9. So here is a table of some naturally occurring fatty acids, and it shows you their structure, the systematic name, the common name, and the carbon skeleton with the colon representation. So dodecanoic acid, that has 12 carbons and no double bonds. So the representation would be 12:0. Tetradecanoic acid has 14 carbons and no double bonds, so that would be 14:0. Now here we have some unsaturated fatty acids. So for this first example, cis-9-hexadecanoic acid, you change the AN to EN to indicate there's a double bond. And then in this representation, that means you have 16 carbons and one double bond. And if we're using the delta numbering, the double bond starts at carbon 9, where we've numbered starting at the carboxyl carbon. If we look down here, this would be a fatty acid with 18 total carbons and 2 double bonds. And the double bonds start at carbons 9 and 12 if we're numbering from the carboxyl carbon. I'm sure you've heard of omega-3 fatty acids because they're essential nutrients. They're very important for human health. Humans need them but cannot synthesize them. And omega-3 fatty acids are called that because they're using the omega numbering system, and the double bond starts at carbon 3 from the non-carboxyl end. So for example, this is an example of an omega-3 fatty acid. So notice with the omega numbering, we start from this end, not the carboxyl end, so that would be 1, 2, 3, and we've got a double bond at 3. The solubility of a fatty acid decreases as the hydrocarbon chain length increases, and I mean the solubility in water. And for melting point, that decreases as the chain length decreases because there are less carbons, and it decreases as the number of double bonds increase. And this can be seen with a comparison of olive oil, butter, and beef fat. Olive oil is an unsaturated fatty acid, so the presence of the double bond decreases the melting point, and therefore it's a liquid at room temperature. Butter, on the other hand, contains saturated fatty acids, which don't have double bonds and therefore have a higher melting point, so it's a soft solid at room temperature. And then beef fat has a greater percentage of saturated fatty acids, which makes it a hard solid at room temperature. The saturated fatty acid chain tends to adopt extended conformations, whereas unsaturated fatty acids, they have a kink in the chain due to that double bond, so they do not adopt extended conformations. As a result of this, whether they're extended or they're kinked, it affects how they pack and how orderly they can pack. So saturated fatty acids pack in a fairly orderly way, shown here, whereas unsaturated fatty acids pack less orderly. And therefore, the greater ordered saturated fatty acids require more thermal energy to disrupt. Therefore, they will have a higher melting point. Versus unsaturated cis fatty acids, these aren't nearly as ordered, so it takes less thermal energy to break those up. Therefore, they have a lower melting point and tend to be liquids at room temperature. I'm sure you've heard about trans fatty acids, that they are extremely unhealthy. They are formed from partial dehydrogenation of unsaturated fatty acids. And the reason this is done is to increase the shelf life or stability at high temperatures, and also to increase the stability of oils used in cooking. A trans double bond versus the cis double bond allows a given fatty acid to adopt an extended conformation, which allows it to pack more orderly, which then results in a higher melting point. However, consuming trans fats has been shown to increase the risk of cardiovascular disease. The majority of fatty acids in biological systems are found in the form of triacylglycerols. If they're solid, they're called fats. If they're liquid, they're called oils. These triacylglycerols are the primary storage form of lipids, also known as body fat. They are less soluble in water than fatty acids due to the esterification of the carboxylate group. and they are also less dense than water. So here's a picture of the triacylglycerol. So this is glycerol. It has 3 carbons and 3 OH groups. And what happens is the carboxyl end of a fatty acid is connected to each of the 3 carbons. So we convert that carboxyl group of the fatty acid into an ester. That's what's meant by esterification. So an ester has an oxygen bonded to a carbon, not an oxygen bonded to a hydrogen. So here we see the ester groups. We have the 3 ester groups here. Fats provide efficient fuel storage, and they have advantages over polysaccharides. They carry more energy per carbon because they are more reduced. Therefore, when we burn them, we oxidize them and can get more energy. They also carry less water per gram because they are nonpolar. In contrast, glucose and glycogen are useful for short-term energy needs. However, fats are better for long-term energy needs. This picture shows a cross-section of human white adipose tissue, and each cell contains a fat droplet, which is white, so large that it squeezes the nucleus, which is stained red, against the plasma membrane. Triacylglycerols are often stored under the skin in certain animals to help insulate against low temperatures. For example, seals, walruses, penguins, and other warm-blooded polar animals are padded with triacylglycerols, which helps protect them from their very cold environment. Waxes refer to esters of long-chain saturated and unsaturated fatty acids with long-chain alcohols. They are insoluble and have high melting points. So these are not triacylglycerols. These are different. So here we have an ester because we have a carbonyl bonded to an O carbon group. And these have a variety of functions. They're the storage of fuel and plankton. They serve as protection and pliability for hair and skin and vertebrates. They're good for waterproofing of feathers and birds. They offer protection from evaporation in tropical plants, and they're often used by people in lotions, ointments, and polishes. Beeswax is a great example of a wax. It's a mixture of a large number of lipids, including esters of triacontanol and a long-chain alkane, hentiacontane. A honeycomb like that shown here is constructed of beeswax, and it's firm at 25 degrees Celsius and completely impervious to water. And the term wax originates in the Old English wax, meaning the material of the honeycomb. So that describes storage lipids, but let's also talk about membrane lipids. There are 5 main types of membrane lipids: glycerophospholipids, galactolipids and sulfolipids, archaeal tetraether lipids, sphingolipids, and sterols. Glycerophospholipids are the primary constituents of cell membranes. They are composed of 2 fatty acids which form ester linkages with the first and second hydroxyl groups of L-glycerol 3-phosphate. The phosphate group, this group here, is charged at physiological pH. So here we have the general structure of glycerophospholipids. So here we have the glycerol component, here's the phosphate head group, and 2 of the hydroxyl groups of the glycerol are connected to fatty acids through an ester linkage. And notice you could have different fatty acids. So this chain is palmitic acid, and this one is an unsaturated fatty acid, also known as linoleic acid. Here are some examples of glycerophospholipids. The common glycerophospholipids are diacylglycerols linked to head group alcohols through a phosphodiester bond. Phosphatidic acid is the parent compound, and then each derivative is named for the head group alcohol with the prefix phosphadidyl. In cardiolipin, 2 phosphatidic acids share a single glycerol. Phosphatidylcholine is the major component of most eukaryotic cell membranes. Many prokaryotes cannot synthesize this lipid, and their membranes therefore do not contain it. Some glycerophospholipids have ether linkages, and these are common in vertebrate heart tissue, and they are also found in some protozoa and anaerobic bacteria. The function, however, is not well understood, but perhaps it has something to do with the fact that ethers are more resistant to cleavage by common enzymes. An example of an ether lipid would be the plasmalogen, which has an ether-linked alkenyl chain, where most glycerophospholipids would have an ester linkage. So here in pink, we see the ether linkage. So an ether is an oxygen connected to carbons on either side. Another example is the platelets activating factor. This is an aliphatic ether analog of phosphatidylcholine, and you see it has an ether linkage, like the plasmalogen I just showed you, but then it also has an ester linkage to acetic acid, so that's a very short chain there. Because of that very short chain, this tends to be more water-soluble than most glycerophospholipids and plasmalogens. The head group alcohol is ethanolamine in plasmogens and choline in the platelet activating factor. This is going to stimulate the aggregation of blood platelets, and it also plays a role in the mediation of inflammation. Let's go to the next group of membrane lipids called sphingolipids. The backbone here is not a glycerol, but a long-chain amino alcohol sphingosine, which is attached to a fatty acid via an amide linkage rather than an ester linkage. And then a polar head group is connected to sphingosine by a glycosidic or phosphodiester linkage. So here's the general structure of a sphingolipid. So here we have the amide connection of the sphingosine to the fatty acid, and the sphingosine is shown in the pinkish color. And then you have a head group substituent. So depending on what X is, you get a different type of sphingolipid. So if X is H, you get something called a ceramide. You may have in the beauty aisle in your drugstore seen a lot of lotions that contain a high amount of ceramides because they're supposed to be lubricating and moisturizing and good for your skin. Gangliosides are very complex, and they have complex oligosaccharides as the head group substituent. And these shapes and symbols are abbreviations for sugars, as shown in Table 7.1 of Chapter 7. Here is a picture of sphingomyelin, which is abundant in the myelin sheath that surrounds some nerve cells in animals. Notice here that sphingomyelin is structurally similar to phosphatidylcholine. They have the same head group, but the phosphatidylcholine does not have sphingosine in it, and instead is derived from a glycerol phosphate. The blood groups are determined in part by the type of sugars located on the head groups and glycosphingolipids. The structure of the sugar is determined by an expression of specific glycosyl transferases. Individuals with no active glycosyl transferase will have the O antigen. Individuals with a glycosyl transferase that transfers an N-acetylgalactosamine have the A blood group, and individuals with a glycosyl transferase that transfers a galactose group are considered to have the B blood group. So the human blood groups O, A, and B are determined in part by the oligosaccharide head groups of these glycosphingolipids. The same three oligosaccharides are also found attached to certain blood proteins of individuals of blood types O, A, and B respectively. So here with the B, what is the difference here? It's that sugar, which stands for galactose. For A, it's this sugar, which stands for N-acetylgalactosamine. And then O, the O antigen, there is nothing here. Now when it comes to gangliosides, dysfunction in ganglioside recycling leads to a variety of medical disorders. And gangliosides are the most complex sphingolipids, and they have oligosaccharides as their polar head groups. So this shows the pathways for the breakdown of GM1, globoside, and sphingomyelin to ceramide. And a defect in the enzyme hydrolyzing a particular step is indicated by an X. And the disease that results from accumulation of the partial breakdown product is noted. So Tay-Sachs disease results from a defect with this process Gaucher's disease, Niemann-Pick disease, Sandhoff's disease, and Fabry's disease. The next category of membrane lipids is galactolipids. In this, one or two galactose residues are connected by a glycosidic linkage to C3 of a 1,2-diacylglycerol. So here's your glycerol, and then here's your galactose sugar. So it's connected to C3 of the glycerol. And here you have multiple galactose sugars. These make up 70 to 80% of the total membrane lipids of a vascular plant and therefore are probably the most abundant membrane lipids in the biosphere. Membranes in plants also contain sulfolipids, in which you have a sulfonated glucose residue joined to a diacylglycerol. Archaeolipids are in archaea, which are organisms that live in ecological niches with extreme conditions such as high temperatures, low pH, high ionic strength, and they have membrane lipids containing long chains that are branched and linked to each end of a glycerol. So this would be an example of an archaea tetraether lipid. So notice here my ether linkages. And likely the ether linkages versus ester linkages provide for greater stability towards hydrolysis. The final group in membrane lipids is the sterols. These contain a steroid nucleus, which is composed of 4 fused rings, and the hydroxyl group or the polar head is in the A ring. And the tetracycle structure is almost planar. Cholesterol and related sterols are present in the membranes of most eukaryotic cells. They modulate fluidity and permeability. They thicken the plasma membrane. There are no sterols in most bacteria. Mammals obtain cholesterol from food or synthesize it in the liver. Cholesterol bound to proteins is transported to tissues via blood vessels. Many hormones are derivatives of sterols. So steroids are oxidized derivatives of sterols. They have the sterol nucleus, but they lack the alkyl chain. They're more polar than cholesterol, and they're synthesized from cholesterol in gonads and adrenal glands. They are carried through the body in the bloodstream, and many of the steroid hormones are male and female sex hormones. This is a variety of steroid hormones. Testosterone here is the male sex hormone produced in the testes. Estradiol is one of the female sex hormones produced in the ovaries and placenta. Cortisol and aldosterone are hormones synthesized in the cortex of the adrenal gland, and they regulate glucose metabolism and salt excretion. Prednisone and prednisolone are synthetic steroids used as anti-inflammatory agents. And bursinolide is a growth regulator found in vascular plants. Most cells continually degrade and replace their membrane lipids. Phospholipids are degraded by phospholipases, and gangliosides are degraded via a series of enzymatic cleavages. And failure to correctly degrade gangliosides results in a buildup of lipids in lysosomes, a dysfunction categorized as lysosomal storage disorders. Now, the final topic is biologically active lipids. So we've talked about storage lipids, we've talked about 5 types of membrane lipids, so now let's talk about biologically active lipids. They are present in much smaller amounts than storage or membrane lipids, and membrane lipids are sometimes also called structural lipids. They play vital roles as signaling molecules, and they include the lipid-soluble vitamins. The enzymatic oxidation of arachidonic acid yields prostaglandins, which are important in inflammation and fever, thromboxanes, which are important in the formation of blood clots, and leukotrienes, which are important in smooth muscle contraction in the lungs. This is the conjugate base of arachidonic acid. This is what it looks like at physiological pH. The carboxyl group is deprotonated. So a variety of transformations convert it into these other molecules. Vitamin D is produced in the skin by UV irradiation of 7-dehydrocholesterol, which breaks the bond shaded light red here. And in the liver, a hydroxyl group is added at C25. And in the kidney, a second hydroxylation at C1 produces the active hormone, shown here. And this is important for regulating calcium metabolism. Beta-carotene, shown here, is the precursor of vitamin A. Isoprene structural units are set off by dashed red lines. Cleavage of beta-carotene produces two of these molecules, retinal, the Al indicates the aldehyde, which is further reduced to retinol, also known as vitamin A1. Further oxidation of the retinol can produce the retinoic acid, a hormone that regulates gene expression. The all-trans retinol can be oxidized to the cis-aldehyde, which is the visual pigment of rhodopsin. When that's exposed to visible light, the double bond highlighted in blue converts to the trans double bond, which forces a change in the shape of the entire rhodopsin molecule. And this transformation in the rod cell of the vertebrate retina sends an electrical signal to the brain that is the basis of visual transduction. Vitamins E, K, and other lipid quinones are antioxidants, and here are some structural examples. The quinone is this portion of the molecule, and these are just more examples. Finally, polyketides are a diverse family of compounds synthesized similarly to fatty acids, and they are commonly secondary metabolites with a specialized function. So erythromycin is an antibiotic, amphotericin is an antifungal, and lovastatin is a statin drug used in human medicine. In summary, in this chapter we looked at lipids, which are a structurally and functionally diverse class of molecules that are poorly soluble in water. Triacylglycerols are the main storage lipids. The phospholipids are the main constituents of membranes. Sphingolipids play roles in cell recognition. Cholesterol is both a membrane lipid and the precursor of steroid hormones, and some lipids carry signals from cell to cell and from tissue to tissue.